a senior biostatistician and associate director of the clinical trials program in the quantitative sciences unit. We're a group of collaborative data scientists in the QSU is here at Stanford University in the Department of Medicine. And I'm joined by my colleague, Isabel. Would you like to introduce yourself?
Hi, everyone. My name is Isabel Weng. I'm a senior biostatistician in the QSU, and I'm happy to talk with Haley about some COVID project today. Yeah.
Thanks. Yeah. And so I'll start by sharing some experiences I've had with COVID projects and lessons learned in the clinical trials world. And then Isabel will talk about similar concerns and how they've been addressed with observational studies. So I'm going to drive my examples with an example trial. It was actually the first COVID trial that I was involved with. And it was a phase 2, we designed it as a single blind study of a single dose of a drug called Lambda, and we're comparing with placebo in outpatients with mild COVID-19. We actually were fortunate, we were able to end up having it be double blind, and I'll tell you a little bit more about that and why it matters later on. So the timing of this was really, when COVID hit, I think we were all very taken off guard, and for us it happened in work too because things move so fast. Clinical trials are usually kind of like large ships that turn kind of slowly and chug along, but COVID really, we needed fast results and information. And so this trial was designed in March. We began enrollment in April, and we locked our database in September for 120 patients who were followed for a month each. And so that's pretty lightning speed for a clinical trial. And the other thing that is a common thread to some of the COVID trials and that I think even today we're still trying to decide how to best approach is we're learning things constantly, which is wonderful. Like the whole research world and community have pulled together to learn everything we can about COVID. But then that makes, our trials usually, we kind of make some assumptions at the beginning, and, for the most part, we kind of let them run along and do their thing. But with COVID, we have new drugs coming out. The standard of care is changing. There's new information available every day. And so there's like this fire hose of information. And that's a common theme I think you'll see with these challenges that I'll talk about that we've encountered as we've designed trials to study COVID. And if you're interested in this particular study, it's about to appear in Nature Communications, and it's available currently on medRxiv. I like to always start with a few examples and maybe I should say examples of what I mean by the language I'm referring to. So some definitions and to familiarize, even if you're familiar with clinical trials, sometimes people use language in different ways. So I'm going to be focusing on randomized controlled trials where participants are randomly assigned to treatment or comparison groups and then followed over time to examine differences in their outcomes. And sometimes outcomes I'll say as endpoints or, but it's what we're trying to observe and make a claim about. And they're considered the gold standard in clinical research because there's randomization and because participants are followed prospectively, where data collection occurs after participants are enrolled. And the groups, part of the reason it's a gold standard with randomization is that groups are alike in all important aspects, and in theory only differ in group assignment because they're being randomized. As I'll talk about in some of the later slides, in practice that's not quite how it works. But in theory, the two groups should be the same, and the only difference is which arm they're randomized to, and that's why these kinds of trials are so valuable. If it's two arms, usually you'll have one treatment group and one comparison group. Sometimes I'll call the treatment group the intervention group or the active arm. I'll say group or arm. And then they're the people in the study who receive the treatment of interest. And then there's a comparison group or a control group. If there's a placebo, sometimes you might call it a placebo group, or usual care group, or standard of care. So it's really just the people who are not getting the treatment, and instead they're getting usually a placebo or some kind of standard of care. And so this brings us to blinding or masking. When you have a treatment group and a control group, it's best if people don't know which arm somebody's in, because there's a potential for bias, either conscious or subconscious, to enter into either the measurements or how the participants are behaving. In a double-blinded trial, I'm starting at the bottom here, both the investigator and the participant are unaware of their treatment assignment. And so in this case, if you have a double-blinded trial, in the control group, if you see an improvement, that's probably just a placebo effect or something happening by chance. And in the treatment arm, then the additional improvement beyond that placebo effect is really that difference of interest that you're trying to measure in your trial. In a single-blinded study, participants are unaware of their treatment status, but the people who are assessing them and the investigators, they sometimes will know their treatment, the patient's treatment status. And then in an unblinded or open label trial, everyone knows the treatment assignment. There's no blinding or masking. And blinding or masking just means that, somebody doesn't know what the treatment is. And so the placebo effect is just one of the biases that can come in when you, if you're not blinded, because you can imagine, like in a single blinded or unblinded trial, either the participants' expectations or the investigators' expectations could, you might be expecting to see an improvement with having this placebo effect. And so some of the measurements maybe could be, especially if it's something that people are self-reporting, like symptoms, they could turn out to look better just because people are expecting them to look better in the treatment group and not really do the treatment. And that's part of the reason double-blinded trials are really the best. If you can do them, you need to have a matching placebo so that nobody knows what the treatment is. And so in this example Lambda study that I referred to, it was technically single-blinded because we didn't have a matching, it was an injection and we didn't have the matching injection. So the investigators who gave the drug, they knew which drug a participant had received. But even when a trial is single-blinded, we try to keep as many of the investigators blinded to the treatment as possible, because it just minimizes the bias. In general, you want to have as few people unblinded or aware of the treatment as possible. In a double-blinded trial, it's only usually the statisticians and maybe the pharmacists who know what the treatment is. And even then, if you have more than one statistician, it's best if you can keep some of the statisticians blinded for when you're doing the analyses at the end of the day. And then only unblind the statistician after you've done all of the programming and locked the database, and then you can do a run of the analysis where you use the actual treatment arms. And so here's another challenge with COVID-19 is that there was a lot of trials going in the same disease area, and it to a degree that we haven't seen before, where there's a limited number of patients, even with so many people coming down with COVID-19 and being impacted by it. But compared to the number of trials, especially in places that weren't hit really hard by COVID, there was a limit. There was a limited number of participants compared to the number of trials. And what we were concerned about when we designed this study, being single-blinded, we wanted to keep the participants unaware of their treatment status, even though the investigators were, because we're concerned that it was possible that participants might choose to drop out of our study if they found out that they were in the placebo arm and go into another COVID trial to try to get an active treatment there. And so when we designed the study, we designed it so that there would be, we assumed a little bit higher dropout in the control arm, the placebo arm, just because we weren't sure if we were going to be able to have the participants blinded or not. Okay, so the next challenge I wanted to talk about that was pretty specific to COVID was that, you know, it's a new disease, and when we're choosing the endpoints, often we will choose from existing and established endpoints. If you're working in a certain disease area, usually there's common endpoints that are used depending on what you're studying. With COVID, there hadn't, you know, there were no endpoints for COVID. What was done at first was when designing trials, we would select from like similar disease areas. Flu, for example, was common. So Flu Pro is a patient-reported outcome survey where you collect information on symptoms, and that, so I saw some studies designing their endpoint based on a survey similar to Flu Pro, or even using Flu Pro, and then over time, what we saw was, especially for trials that involved inpatients, is there was sort of an evolution of endpoints where more was known about the COVID patients and what to expect, and there was scales that were developed, and you maybe have seen the WHO scales where they range from death at the worst, and then ventilation, different levels of ventilation, and then hospitalized, and then people being discharged, and in those scales, even though they were established pretty early on, there was still some evolution in exactly, you know, what the different levels in those scales were over time. And so one of the things that's a challenge, that you have your endpoints changing over time. Not over time within the study. We want to keep the endpoint the same within a study, but over time, in the course of, you know, trials being designed to study COVID. In the example trial that I was, you know, showed at the beginning, Lambda, it was looking at, it was an outpatient trial, so it was different than a lot of the trials going on at the time, and it's studying the viral load and whether people were shedding virus. And so when we have a few studies actually where we're looking at similar outcomes where they're antivirals, and we want to see how well the treatment improves the time until people stop shedding their virus. And so some key features in general of good endpoints are that you can observe the endpoint. Of course, you want to design your trial in a way where you have a chance to see your treatment take effect. We want the endpoint to align with your research question and the outcome of interest when designing a study. And then we want to minimize the variability. The less noise you have, the better. And so in the example study, we used time until shedding cessation, so the time until we stop seeing active virus. And there were other trials that were using like proportion of participants who had stopped shedding by day 5 or day 7, for example. But we chose not to go with that because we weren't sure what the right time period was. By having time to shedding cessation, we could, you know, even if the amount of time it took to stop shedding was different than we expected, we could still observe the endpoint as long as it was within our follow-up period of 28 days. And so I think one of the challenges with COVID was, you know, there was a question of what are we measuring, what should we be measuring, and then of course, how do we measure it? And one of the things we did in our group, the QSU, was we created a registry of endpoints so that we could see on clinicaltrials.gov what other endpoints were people using, and see, you know, get ideas and see what seemed to be working for others and learn from what others were doing. Okay, and I think the one big challenge that I think this is one of the things that we're starting to deal with a little bit more now is, what should the interim analyses look like? The early stopping considerations were different at first in COVID because little was known about the disease. There weren't any treatment options available, particularly for outpatients, which is more most of our trials are. And there was a general need for information. So there wasn't much of an incentive to stop unless we saw a really, some kind of signal of harm. If we saw that there was harm with the treatment, of course, we had a stopping rule to, and a DSMB, a Data and Safety Monitoring Board, to watch for any potential harm. But if it looked like the treatment was futile and not working, or if some of our assumptions in the design were off, we wanted to continue the trial because there was a real need for information. And what we've done over time with those participants is we've also looked at secondary endpoints like their symptomology and learn from that, and are able to report on COVID, symptoms over time, the natural history, because, any information is really useful. But now there's more treatment options available, even in outpatients. There's some other options, not as many as there are for inpatients, and there's still not a good suite of treatment options, but there are some treatment options now for outpatients, and that need for information isn't quite as dire as it was at first when we didn't know anything about the disease. And so now it's, the balance has shifted a little more, where we're more likely to, we've built in futility analyses where we're going to stop enrollment if we see that it doesn't seem like it's a promising disease or a promising intervention. And then the other piece is that because there's, I mentioned before the fire hose of information where there's just so much being learned even still, between the time we design our study and by the time we have our interim analysis and our final database lock, there might be a lot learned. And so we've started to evaluate whether we need to be doing more at the interim analyses and evaluating the design assumptions using aggregate data at the interim timing. And in a trial we're designing now, we are considering doing a sample size re-estimation at the interim analysis to account for the potential that maybe our assumptions about how many people are receiving other treatments, how many outpatients are receiving other treatments, maybe off, and we may also be, it's a way to evaluate whether our assumptions about the in the initial power and sample size calculations are sort of close, or if they're pretty far off, and so we're starting to the way the interim analyses look are changing over time. And what's challenging here is we don't know what we don't know, right? Like when we were designing the study, we have things that we think we, maybe are not so sure about and we want to evaluate at the interim, but we don't, it might end up being at the time of the interim, it's something else that's seems like it's potentially problematic. And so I think what's challenging is just there's so little that's known and there's so much that's still being learned that it makes it tricky to design these studies. Just on the last note, I wanted to leave you with a couple of takeaways when thinking about designing trials, especially when either they're studying COVID or even trials that aren't studying COVID now. There's so many unknowns because there's fewer patients coming into the hospital. We're seeing fewer, infectious diseases. We have some pneumonia studies where we're just nobody getting pneumonia these days. And so, and just in all the trials now, I think we need to be careful and mindful of how COVID's impacting them. And I think you want to choose not just an endpoint, probably any design feature. You want to choose something that's robust to the unknowns and try to, like I gave the example earlier of time to cessation versus viral shedding on day 7. It does, time to cessation is not reliant on the actual time that people are stopping shedding, and so it's a little bit more robust. And you can build some of these complexities in about the unknown assumptions, but it also increases the design time. And in COVID, we want to be getting trials out very fast, so there's a trade-off there. And then it's always good to include any mitigating strategies or sensitivity analyses in your study design and statistical analysis plans. Common things are like log transforming your data if your outcome's non-normal, or you expect to have maybe some non-adherence in patients. And that's especially true when you're designing a study during this time of the COVID pandemic. So that's it. Enter questions you have in the chat if you haven't already, and I'll turn it over to Isabel.
Following Haley's discussion on clinical trial for COVID, I'm going to talk about some topic issues in COVID research when we are designing an observational study, especially nowadays we're using data from electronic health records to do those observational studies. When we talk about real-world data, it's usually referred to the following main group of data sources per FDA's definition. Today, my workshop topic will mainly focus on the electronic health records, and which is the main and the emerging hot topic in the real-world evidence-based research. Then what's EHR? According to healthit.gov, it's just a digital version of patient's paper chart, but it's real-time patient-centered records that make information available instantly and securely to authorized users. There are some key distinguished features of EHR data that differ it from research data. First of all, the data are generated per clinical purpose instead of research purpose. As a result, there's no formal and robust data quality checkups conducted in EHR. Another limitation of EHR is that although it could be longitudinal, the follow-up frequency and the spacing could vary across patients, which is likely to be in a non-random fashion and driven by the need of patient care. Then let's talk about the study design consideration. I think this is the favorite pyramid which shows the hierarchy of evidence that Haley and I both like. And for me, our RCT is usually on the top, and on top of that is the systematic reviews of all the RCT. But for researchers use EHR, it's likely to fit in the cohort study bucket, and whereas I think it's closer to the retrospective cohort study, and patient data is followed over time only for the purpose of clinical care rather than research testing a research question. As similar to other types of study, I think the key study design consideration, which is the first step, is to think about what's your relevant question to ask. This looks very straightforward and at most time requires a comprehensive review of the literature of the relevant disease area and the topics. However, for COVID-19 research, we face some unique challenges. As it's a new disease, the scientists have very limited knowledge at the beginning. It becomes very hard to have a most relevant research question for our clinical collaborators since there are too much to be studied. As Haley mentioned, the landscape could evolve very quickly over time. And we, as their data partner, it will be helpful to first have a scientific conversation with them and narrow down, prioritize what to look at first. Then we can help them assess the feasibility using the existing data. With the primary research question being specified, we will next like to design a study to ensure robust internal validity. That internal validity is it just means like you have the right data and the design to answer your research question. This requires us to have appropriate method to identify the most relevant cohort of interest. We need also think about the potential source of selection bias and a non-response bias for EHR, which is not usually a huge issue for randomized trial prospective study if they're well designed. In addition to that, we should take into account the robustness of the measurement in EHR during our design. As I said, there's no QC step for EHR data. It's just the real-time data that generated according to patient care. And in terms of ways of handling missing data, there's opportunity to utilize different components within the EHR as well as linking external data sources to minimize the missing data and measurement errors. For COVID-19 researchers, there's also unique challenges at the beginning when we're trying to define a COVID-19 patients, because there's no diagnosis codes available at the beginning. That's a new disease, there's no diagnosis code. And also the clinical guidelines for who are the COVID patient are not available and consistent at initial stage. Furthermore, the accuracy of the lab tests are not ideal for us to use, especially when it's at the beginning of the COVID pandemic, say like April and May 2020 last year. Next, I will talk about how we will define a real world outcome when we use data like EHR. There's typically several big buckets of outcomes that are very likely to be robustly measured in EHR for clinical diagnosis or treatment purpose or operational purpose. This could include those buckets outcomes like patient's benefit and side effects for specific disease or treatment, institutional costs and process efficiency, and sometimes patients and provider satisfaction. For COVID-19 research, our classic institutional quality improvement outcomes such as length of stay, mortality may be relevant, but not necessary to be ideal direct measures to improve patient care. Similar to our CTS, Haley mentioned, the most challenging piece is just to identify the right patient level outcome that we can measure. That's another unique challenge is we cannot measure anything in EHR. We can just use what's available there to measure patient level outcome. This is also because we know very little about the disease initially, and there's there's a landscape change over time. The outcome could change over time. So that's the most challenging piece when you wanted to design a good outcome for COVID-19, especially using EHR. Also, we need to also consider the population heterogeneity of our patient population and consider whether the outcome is relevant to our local institution. Lastly, again, we should keep in mind the longitudinal nature of EHR have a clear understanding of the difference in its data generation process comparing to prospective cohort study in RCT. And there's usually be selection bias that introduced, as I mentioned, due to the differential number spacing and follow-up visit for all researchers using EHR, because people's visits or follow-up visits is driven by whether they wanted to get a clinical care, which it may or may not be associated with outcome or the intervention they receive. Next, I'd like to talk about some of the statistical consideration. Haley might already mention we usually have an SAP for all the clinical trial, but I will recommend it also implement an SAP prior to data collection, even you're doing an observational study and using data such as data from the EHR. Unique to EHR, I think there are some key perspective to be included in your SAP. One is, of course, you need to outline the primary statistical method for your primary outcome evaluation, and you need to define your primary outcome clearly in your SAP. And you need to think about the missing data and the underlying assumption, especially for your missing outcomes, which is likely to introduce the selection bias. Also, you need to think about the potential impact of differential follow-ups and have some sensitivity analysis to address that. I think another point is very important is usually it might be helpful to give the clinical collaborators some guidelines on how to interpret the results under the EHR context and have some guidelines of what's the typical limitation will be with your study design. One important limitation that researchers are often concerned about EHR is the missing data outcome problem. Specifically, both confounding factor and outcome could be missing. And usually the traditional missing data imputation techniques, such as mutable imputation, which rely on assumption of missing at random, might not hold true in the EHR data. For COVID-19, a timely reporting system specific to COVID within EHR is needed. this is a little bit challenging at the beginning because it relies on collaborative efforts across all the team within the institution in order to ensure the data completeness and the data quality and accuracy. How are we gonna handle missing data? The first solution is always considered to get the data from somewhere else. This may be possible for EHR because it's a humongous data system that capture a lot of information on patients at different places and their different encounters with the healthcare system. It's very likely that a single variable could be collected in multiple places within the EHR that can make them complement one of each other. In order to identify all those relevant information within your institutional EHR, a test data extraction should be considered in the study protocol. This will probably help you discover any missing data related problems early and make the data extraction process more smoothly and efficiently after the study implementation. An alternative way to get data is try to find some kind of surrogate information that you can use to link to the external data in order to capture what you want. For example, you can use zip code to link census data in order to get a sense of patient social demographic, social economic status, such as median income, household number. Although that's within the zip, but it's usually sufficient if just the purpose is just to incorporate them as a confounding factor to adjust in a model. So say you try very hard and then still get some of the missing data. Therefore, there will be those fancy statistical imputation methods that come to play a role. However, we should always have a good idea of the underlying assumption of each imputation method and check how those assumptions align with your data before implementation. And I think it will always not hurt if you just can have sensitivity analysis outlined in the ASP that tries a variety of imputation methods to check the robustness of your primary findings. And I listed some of the options you can do under different missing data assumptions. In the SIP, you should have a very clear and straightforward the statistical modeling. And I will think sometimes I will list it out the equation just for if there's other statistician want to look to it, and that will be straightforward for them to implement your model again. In addition to that, as I said, and Haley might say, we need to outline sensitivity plan, like such as we wanted to try alternative modeling approach like GE versus mixed effect model, which is you probably listed as your primary, but you can have GE as your sensitivity analysis. Also, there's a unique challenge of EHR that I mentioned before, is the differential follow-up and the follow-up problems. The result of that is you will get a selection bias that will probably hurt your internal validity, especially when the spacing and the number of follow-ups is dependent on how you measure the outcomes and how people get a intervention or not. To address that, you could try to specify sensitivity analysis under a variety of assumptions, such as like you can assume some kind of worst case scenario or apply matching techniques on the number and the spacing of the follow-ups to show that your comparison group have similar follow-up schedules. It's all Another important thing that we as statisticians should constantly remind our clinical collaborators are to include all the limitations of the study design and statistical method into the discussion section of the paper. This requires us to collaborate with them to do reviews of the final manuscript. Also, we need to be very transparent on all the sensitivity analysis we have done and discuss all the potential bias that could occur with our study thoroughly into the discussion section. I also recommended to just have some discussion on the external validity and the generalizability, because data from your institutional EHR are very likely to be very institutional specific, which may or may not be relevant to the other population. You need to have your conclusion within the scope of the data you have. For COVID-19 research, as mentioned, and Haley probably mentioned again, it's the most challenging part is the landscape changed very quickly, and there's a very short turnaround time, given the quick emergence of the external evidence that sometimes you design a study and after a while, others will find similar finding and your data might not be so relevant. So there's a quick turnaround time for you to submit manuscript, have your result disseminated. Here is just a summary of the challenge I've already mentioned about COVID-19 research using EHR data. There's a real world example that I'd like to briefly talk about. Basically, in responding to the surge of the COVID-19 cases, Stanford Department of Medicine initiated a weekly reporting effort of the COVID-19 hospitalization population during the grand round, starting from May 2020. Here's just a snapshot of the information for our department, which is available to the public on YouTube. And then the primary purpose of it is just to have a weekly reporting and use the data to help clinicians to understand the most up-to-date landscape of their tested positivity, characteristics of the hospitalized patient, and their outcome within our system. The request sounds to be very straightforward. However, when we think about it carefully and try to implement a real-time reporting system, we found a lot of challenges. Firstly, it's quite hard to define a COVID-19 hospitalization cohort at the beginning for the reason I mentioned. We don't have a diagnosis code. We have some kind of real-world data from the EHR, which is not clean. As we all know about EHR, the most updated data could usually not be so good. And the system is very dynamic. There's intermediate lab result that's existing in the EHR. There could be lag in the data entry, and the lag could be different for different components. Say you have the hospitalization data lag for 4 days, but the lab data is more in real time in 1 day. We need to take into account all those dynamic features of the EHR system. We wanted to have a robust reporting of your number. Also, because of those dynamic features, if you wanted to do a QC, usually there will be cases that data for the same historic period could change if you try to report it at different time. Sometimes it's acceptable, sometimes it's not, such as deaths. You cannot see people died at an earlier time and they disappear later. Then that will be a problem you need to have further investigation. This also means if you want to incorporate a QC plan over time with EHR for reporting purpose, it's quite challenging. All those nuances could be only learned if there's opportunity to collaborate with our clinical collaborators and our data engineer who have depth knowledge on how the EHR works within our institution. As I mentioned, there's a challenge that we cannot know who are the cases, who are the hospitalized cases, because we don't have diagnosis code, we don't have clinical guidance, and the accuracy of the lab is not ideal. So our decision is, at that time, we think our clinical collaborator thinks the RT-PCR result is the gold standard. So we will use it to identify our cases and hospitalized patients. But what time of the RT-PCR report is used, then there's a decision of the time point. So at that time, we decided we only account for the initial hospitalization, whereas the admission date is within, say, 15 days of the most recent validated RT-PCR positive results. And with this approach, although there's limitation, but we could conduct sensitivity analysis. And also we did some independent chart review on our cases initially to ensure our definition have best performance and give us, this also gives us an opportunity to identify any potential limitation and maybe revise the definition accordingly if necessary through chart review. So from the chart review and of the case, our initial cases, we found that our definition might capture some incidental hospitalized patient who were hospitalized for other reasons. Say a woman got pregnant and then go to hospital for their to deliver the baby, and they at that time they did not have the COVID symptom, but there's a require of a RT-PCR test at admission, and they were incidentally to be found that they have a positive result. For those patients, they are included, but after talking about this, I think our collaborator are fine with including those patients. And as long as we have our definition on the limitation clearly stated, it will help us to understand what's our cohort and understanding the relationship of the potential outcome. For outcome, there's like such simple outcome like transfer or admit to ICU. There's a not ideal single variable that you can use. What we do, we just incorporated multiple sources for data from the EHR in order to minimize the risk of categorizing the ICU patient incorrectly. Classic outcome that people are always looking for COVID-19 reporting is the positivity rate and hospitalization rate. As shown by the right, for the Santa Clara dashboard, they have a really nice graph of test positivity over time. This is very interesting to our clinical collaborator, and they wanted to know their local rates within the hospital. However, we realized that we're probably not able to estimate those measures within our institutional EHR data. The reason is Stanford Healthcare is an open system. Those patients who come here for tests and test positive might not necessarily seek care within the system, so they are not necessarily go to Stanford for their hospitalization. And at the beginning, Stanford is the main site that have the testing available. So everybody are only available to get a test from Stanford. In other words, that we don't have a good way to capture our denominator for those proportions, so we are not able to get a reliable estimate accordingly. Similarly, you probably cannot get any estimation remission rate, but it's not so less likely to be underestimated as those positivity rate and hospitalization rate as for the reason I mentioned below. On the other hand, what we can do is to report the crew counts, and that will give us a good sense of the trend over time, how it compares to the county data, state data, and the national trends. Some illustration of the case numbers and the hospitalizing numbers are shown here. We really see a very similar trends for our system and the county. Here are the lessons learned. I think first we need to think about carefully on your algorithm of the cohort definition. And that's an iterative process that you need to consult, collaborate with the end users and the developers. Also, you need to think about the data points from the multiple sources within EHR in order to handle the missing and the reporting outcome measure. We might not able to estimate an outcome that require longitudinal follow-up, especially when you are using a EHR data from the open healthcare system. That's all of my discussion today. I'm happy to answer any question, and please, I will stay on the live chat and address all the comments and questions that you have. Thank you.